Dr. Simran Kaur was a graduate student in Dr. John Christodoulou's lab, and she studied KIF1A mutations. A couple of years ago, they received a grant from KIF1A.org to start studying hand-related mutations. And since then, we just continue to see SIM expand and expand the program, and not just expand it, but expand it in a way that keeps the patients in mind. When SIM reaches out to me, it is always a few questions. Has this work been done? Is it duplicative or helpful? Who in your network can I talk to make sure that it's even more effective? And so over the course of this talk, Sim will be introducing a lot of ways in which she's expanded the program of researching KIF1A and looking for cures for CAND. And I hope that you all be excited by all the possibilities that are looming, and I just can't wait to hear her tell you it in her own words. So Dr. Simran Kaur, if you will.
First of all, thank you so much. Thank you for this warm welcome. And just meeting with many of you in person, and even if I didn't get a chance to say hello to you, just stepping back and just observing the dynamics of this close-knit community, it's such a privilege to be part of it. So thank you so much for accepting me as one of you as a member in KIF1A.org. So again, I would like to say a big thank you to KIF1A.org, who first of all gave me this opportunity and the platform to explore CAND. They gave me the opportunity to meet with other clinicians, scientists, families who have got this common mission to know more about CAND. So, of course, and all the KIF1A.org research network members, they have been there to support me in this journey. Whenever I've got a question, I would ask KIF1A.org, Dominic, Dylan, can you please put me in touch with somebody who could help me do this? So if there is a question, I'm sure there's going to be an answer, but I have to find the right people who could support me. So a big shout out to KIF1A.org and all of you. A huge, huge thank you. Dr. Chung has given such a lovely introduction about what CAND is and what they have observed in the natural history studies. But I'm sure there will be many families who are new to this community. So I'll give a very quick introduction about what we know about CAND. It's an ultra-rare brain disorder that we know of at the moment affects nearly 500 individuals worldwide. Now, we believe this is a significant underrepresentation of the actual numbers of CAN in our community, maybe because of two reasons. First, they are not genetically diagnosed yet because of the limited accessibility of the genetic screening, or they are misdiagnosed as other conditions, mainly the cerebral palsy that Dr. Chung had briefly discussed in her presentation. It's a brain disorder, but it affects multiple organs within the body. And it can come as some of these symptoms, and they might change over the time, like how Dr. Chung was talking about. So I won't focus too much on this slide at the moment. I thought I'll give you a little introduction about what neurons and how does it function too, just to kind of set the scene for the talk. CAND it's a brain disorder, and it's caused due to the changes in the gene, which is called KIF1A. KIF1A is mostly expressed in the brain cells called neurons, and this is typically how the neuron looks like. So we've got the dendrite structures that basically receive the message from the neighboring cells. We have got this large cell body. It's like a factory of the proteins where most of the things get manufactured, and they have to travel through the long axons towards the neurite terminals in order to pass the message across to the next cell. Now, this is done by specialized proteins. They're called motor proteins, one of which is KIF1A. So KIF1A, it's a dimer. So when we call it as a dimer, it's basically two wires which are twisted around each other, like in the electrical wire, and it moves on the long highways within the cell, what we call as microtubules, which you can see here stepwise, one step at a time, to carry those bulky cargoes which are getting manufactured in the factory, that is the cell body, towards the neurite terminal to pass the message across to the next cell. Now, within our cell, each gene makes one protein. So a normal KIF1A gene makes a normal KIF1A protein, and that results in a normal movement of cargoes. However, because of small spelling mistakes within the sequence that codes for the KIF1A gene, what we call as a variant, and now this is a variant gene, it makes variant KIF1A protein that results in abnormal cargoes. Now, the variants, they can come throughout the gene, like what Dr. Chung has presented. So the whole part of the protein and the gene, they are susceptible for having these small spelling mistakes. But most of the research has shown that a major proportion of these variants, they're found in the 2 feet that walks on the microtubules. And just for the simplicity, I have noted the variant as a red star here. Now, again, if there is an abnormal movement, what's going to happen ultimately? Cargoes are not going to move. There is no communication between the cell. And what it does is basically starts a cascade of other events that starts to slow down the activity within the neurons, affecting the overall function of our CAN individuals. Now, the variants, like we know, they affect the function of the protein, mostly resulting in loss of KIF1A function, but they do so in different ways. They can either result in, they interfere with the binding of the KIF1A protein with those long highways called microtubules, such that they fall off from those highways, or they interfere with the speed with which the KIF1A motor is moving along the microtubules, either resulting in a slow movement or no movement at all. Now, what we are doing together, KIF1A.org and all the research network members and the community, we want to restore this abnormal movement back to the normal such that we can improve the quality of the life of the CAN individuals. Now, I'm going to give you a little bit introduction about the CAN research program that we are building in collaboration with KIF1A.org and its various research network members. Our topmost priority is to engage with the CAN families, to know about their perspectives, to know about their priorities, so that we can convert the problems that they face on a daily basis into the projects that we can study in the lab to help address some of those issues. Now, with the very kind and generous support from KIF1A.org, Australia's National Health and Medical Research Council, and Murdoch Children Research Institute, where my lab is based, I'm quite privileged to say that we have developed Australia's first research program that solely focuses on the KIF1A research. It has got 5 pillars. The first 2 pillars is basically to model CAN in a dish in the lab, so that we can study more about what is happening within the cells when a person has got a CAN. So that is basically modeling CAN in the dish and understanding its background biology. Why do we want to do that? So that we can convert this knowledge to develop therapies and treatments to benefit CAN individuals. Along with that, we are also working with other team members around the world to improve the diagnosis and basically the early diagnosis for the CAN. And advocacy is something which is very close to us. We want to let the community know what CAN is, what its complications are, and why do we need such an urgent care and urgent treatment for CAN. So let's start off with the pillar first. Like I said, CAN is a brain disorder. But this is a tissue which is not so much easily accessible for us, the scientists, to work in the lab. So what we try to do is we try to find alternate ways of studying KIF1A in the dish in the lab. So here I'm going to introduce you with 2 main techniques that we are using to build those models, like how we are studying KIF1A in our labs back in Australia. So the first model is called COS7 cell-based model. COS7 cells, they are the monkey kidney cells. They're very versatile, they're very easy to grow, and most of the KIF1A community like working with them. But because they're kidney cells, they don't have KIF1A in them. So what we try to do is we try to make some artificial KIF1A within those cells. We are not working with the full length. We're only working just a part of the protein, the 2 feet, which are more susceptible for the variations to occur. And that's the part which is mostly affected, as we know of at the moment, in the KIF1A protein. So if I show you here, this is a part that I'm talking about. So 2 feet, they're now tagged with an artificial tag, which basically emits out green light on a microscope. We want this because otherwise, how we are going to know where KIF1A is going? So it basically helps in visualizing the position and its trafficking along the peripheries of the cell. So this is a normal KIF1A. So you can see here the schematic of a COS7 cell. In case of a normal KIF1A, like it has a tendency to move efficiently on the microtubules, it starts to kind of start accumulating at the periphery of the cells. Whereas in case of the variant KIF1A, we have found that because the walking pattern is interrupted, so it mostly is stuck within the center of the cell. Now, on the right-hand side, you can see those two beautiful images, which is taken from special microscopes. In this, the green is the green light that I was talking about. It's coming from KIF1A. So green represents KIF1A localization. Red represents the rows on which it moves, that's the microtubules, and the blue is the nucleus. Now, these are the time-lapse movies. So what we try to do is we try to take the still images. Along with that, we want to see how dynamic is the action of KIF1A. So we try to take movies within the cell at a particular interval of time over the 3 days period and try to see how KIF1A is behaving. So like you see on the left-hand side, this is a video captured with the KIF1A COS7 cells that is expressing the normal KIF1A. So as you can appreciate, the KIF1A starts, if I play the movie again, so you can see the KIF1A is getting more accumulated at the peripheries of the cell. It's mostly like the lightning happening. But more closely, if you pay attention, it's basically forcing the cell to make those long neurite-like structures within the cell. So somehow KIF1A has got a role with that, which we want to explore more. On the right-hand side, you can see a movie of the COS7 cells, which is making the defective KIF1A protein. It's mostly localizing the cell body, and the cells, they're not doing much in terms of making those projections to reach out to the next cells that they have got. Before I move on to the next model, I would like to say that this is a model that we are using for the high throughput drug screening of small molecules so that we can find out, is it an FDA-approved drug which can act on KIF1A? I'm going to talk more about it later, but let me come back to the models, the first pillar that we were talking about. So the second model, it's about the induced pluripotent stem cells. COS7 cells, they're great. They work very good if you want like a very rapid experiments within the lab. But we do need some models which is closely representing the brain models derived directly from the patients. I know it's a bit of a mouthful, so we'll say the short name for now. It is called the iPSCs. And we are going to have a lot of other researchers which will be talking about the iPSCs. So a little introduction about how do we get them. So we start off the iPSCs. We can, in a very simple terms, we can think of these are the special stem cells which can be derived from the blood cells or the patient's skin cells. We do a little magic in the lab and we reverse the clock so that they again go back to the state where they have got this potency to make any cell type within the body. For our interest, what we want to do is we want to look at neurons as well as the small mini brain organoids that I'm going to talk you through in the next slide. But at the moment, what I want to say is, with these iPSCs, we have got 6 different variants at the moment, which we are focusing on solely because we had access to these iPSCs from these particular variants. With time, if more iPSCs are available, we would be shifting our gears to study more variants and see how the KIF1A is doing in those cells. So like I said, we are using the iPSCs from CAN individuals to convert them into 2-dimensional neurons or 3-dimensional brain organoids. Now, brain organoids, they are like the mini brains in a dish. So after culturing them for about 5 to 6 months in the vessels, in a special media, we get a structure which closely represents the structure as well as the function of a human brain. So they make a very versatile and personalized model for us to study CAN in a dish. Coming ahead for understanding the biology behind CAN. Now we're working very closely with KIF1A.org to know the key problems that the CAN community is facing so that while we are working to address the root cause of CAN to fix the KIF1A, which are the areas that we can focus to improve the quality of life, like Dr. Chung was saying, care until we cure. So there are the three main areas that we are focusing on at the moment. It's the treatment-resistant epilepsies that affects nearly about 5 in 12 children, and vision as well as speech. So these are the things which are most commonly observed in almost all of the affected CAN individuals. Now, in order to understand the mechanisms behind the severe seizures and to find the targeted therapies, we have combined together an excellent team who is going to put their brains and minds together to understand a bit better about these treatment-resistant epilepsies. Again, our ultimate aim is to find the targeted treatment. Now, you would see a lot of familiar faces. So we've got a lot of our KIF1A Research Network members who are part of this team, and I'm very thankful for them. The other thing that I want to focus here is, this is probably the first time Australian National Health Medical Research Council has supported a grant or supported a funding to execute a grant which is for KIF1A solely. So it's a huge success. And just talking about the numbers, only 6% of the grants were successful in the round. So this gives us a huge, huge confidence that yes, the Australian government and the community there is hearing us out, that yes, the urgent medical treatment and the therapies are important for rare diseases, especially for CAN at this stage, which is a huge win. So before I talk about the methodology of how we are going to work on this particular grant, I would like to say thank you to our two amazing research assistants in the lab. It's Kelly Gao and Tim Sikora, who are doing different aspects of the projects in parallel so we can save on time in running this particular project. So very basically, once again, what we're doing is we are growing the cortical brain organoids, or just simply brain organoids, and the neurons from the patient iPSCs, which are currently in culture. And the plan is by the end of the year, once these organoids mature, we are going to put them onto these special plates, which has got some electrical wires at the bottom. We call these plates as multi-electron arrays, or for short, let's call them MIAs. So what it helps us to it helps us to record the activity which is going on these artificial mini brains in a dish, so that we can see those waveforms, and then we can capture the waveforms from the CAN individual brain organoids and their counter control or the wild type organoids. Now, what we want to do eventually is we want to compare it with the clinical EEG data that Dr. Chung and Dr. Bain, they are collecting as part of their studies, and we want to find out, does the electrical activity that we observe in the organoids, it matches with the EEG waveforms. So what will be the outcome? So with this, what we are going to do is we are going to create an in vitro seizure prediction model, so that it can help us to understand if we come across a particular KIF1A variant, what can we expect for the seizures just based upon those artificial brain organoids that we grow in the lab. At the same time, what we are doing is we are developing, as I said, the high throughput approaches to find the FDA approved drugs. Once drug is identified, we are going to treat the brain organoids, and we're going to see if it does any changes to the electrical activity to bring it back to the normal. More so, what we want to do is if the drug is doing this magic thing in the brain organoid, we want to look at more carefully at the biology. We will do a different number of sets of experiments to know about the pathways, the networks with which that drug is working, so that again, it gives us the power of finding other possible FDA approved drugs which can act on those particular pathways to, again, with the common aim of finding the treatments for these resistant epilepsies. Now, the second priority that we are focusing on at the moment is the vision. Now, using the power of clinical information, which will be available through KIF1A.org's research network members, and as well as developing some artificial in vitro models, we want to understand more about vision, what is going on, and then what are the mechanisms behind the different vision problems that the CAN individuals they are encountering. So for this, we have extended our collaboration with Dr. Anay Gonzalez Cordero, who is one of a very remarkable scientists in the world. What she does is she grows mini eyes which are connected with mini brains. We call it as the retinal brain organoids, and you can see here there's a little part of the brain which is growing in culture with little part of the retina of the eye. And the two parts, they're connected with the nerve-like axonal projections, which is basically the other term for the optic nerve, which is going, what it is doing, it's basically in the retina, the photoreceptor cells, they get the information, they convert it into the signal, which then goes to the optic nerve to the brain to comprehend that particular image. So you want to understand more about it, like what happens in the CAN individual, what are the pathways which get affected, and how is optic nerve atrophy is exhibiting in these models that we are growing in terms of the eye and the brain organoids. At the same time, we are also collaborating with Dr. Catlett's group. Thank you so much to them, to understand this phenotype a bit better using the CAN models that they're developing for the KIF1A community. And what we want to do is we want to see if the patterns that we are observing in terms of the retina or the eye-brain organoids, they matches with our results from the mouse models. Again, this will give us more confidence about knowing the biology and then hopefully finding out is there any drug or is there any treatment that can fix the eye problems or the optic nerve problems? Moving forward, we do want to improve the quality of life. As Dr. Chung was saying, there is a lot of times that the individuals are not able to comprehend the issues that they're going through. So speech is something that we shifted a little bit of gears to understand a bit more, like how the speech and language is affected in the CAN community as compared with time, and of course as compared to their other age-matched individuals. So for this, we are working together with Professor Angela Morgan. So she works very closely with the rare disease communities, actually a number of genetic disorders, to know about the speech and language difficulties in order to again improve the quality of life. So what she has done is she has developed this remarkable pipeline to know more about speech. And now, in terms of the pipeline, it's very inclusive of the children, individuals, and families speaking with different languages, as well as the minimally verbal as well as non-verbal individuals as well. So Professor Angela Morgan, along with her PhD student Lottie Morrison, is very, very keen to engage with the community to know about more like what are the expectations of the community and how can we co-design this project so that we can benefit the KIF1A community in future. So what will be the outcomes? So the results from these studies can help in prognostic counseling. It can help us to provide data which can help for the early interventions in terms of accessing the speech therapist, that then they can form some targeted therapies that can benefit the CAN individuals. Along with that, it gives us information about campaigning to the insurance companies to support these therapies. And of course, like if possible, then we are going to use these as the measures for the clinical trial outcomes as well. Now coming to the pillar 3, which is focused on finding the treatments for the CAN. So our strategy to find the small molecules or what we call as a drugs for CAN is based upon drug repurposing. It's basically another very simplified version. We call it as a drug recycling. So there are a lot of FDA approved drugs that are already there in the market. So what we want to do is we want to look it up more carefully and see, does any of the drug actually works for KIF1A or not? So this can save a lot of time, resources, as well as it's more time friendly and economical as compared to going ahead with what we call as a de novo or the new drug development, which is on the left. So it saves us a lot of time in that way. So again, it can be done in a couple of different ways. There is low throughput, which is basically finding and looking at one drug at a time, or we can do this as a high throughput in which we can make use of the robotic system that can actually look at thousands of drugs in one go, really saving us time when we are looking for the small molecules for CAN. And on the video on the right, you can see the high throughput drug screening robot in action in our facility, which is basically treating the different cells at one time with the possible drugs. So it's very, 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 very quick. Now, without going too much details about the background for developing models and developing this pipeline, we've got some preliminary results that we have created with the support of Dr. Alejandro Hidalgo Gonzalez and Dr. Henry Beetham, in which they helped us to look some of the tool compounds and to look up their action on the green light, which is attached to the KIF1A. So on the graph on the right, you can see a lot of curves moving up. Basically, we have found few tool compounds, which basically helps in increasing the levels of the green light in a very dose-dependent manner. Now, which is quite encouraging for us to see. We don't know whether this means anything in the clinical context, but yes, this is something that we would like to explore more, that just increasing that the expression of the green light, does it parallels with increasing the expression of the KIF1A or not? And if so, will it help to rescue any of the functions? Now, along with looking at the small molecules for CAND, we do want to address the root cause for improving or for fixing KIF1A. And for that, we have joined hands with Associate Professor Wendy Gold to explore her idea about the gene replacement therapy for CAND. Now, the idea is solely based on the prescription gene therapy, which is currently available for spinal muscular atrophy or SMA. The name is Zolgensma. The approach is likely going to be similar, and we want to see if we are going to have some effect using that particular approach for CAN individuals. So briefly, what is the idea? What we are going to do is we are going to create this artificial normal KIF1A gene. We are going to pack it as a package, and the package here is AAV9. It's a bit of a mouthful again, but what these things do is, first of all, they're the virus, but they're clinically safe. They don't make people sick. But what they help is they help to take that normal KIF1A into the cells of the individual, such that the KIF1A starts expressing, the normal KIF1A starts expressing. So this is an idea, and we are lucky to have some support from Murdoch Research Institute to explore this idea and see if this is going to be any clinical significance or not. And we have got our honor student, Grace Lim, who will be doing this project this year. So of course, we'll have some news next when we are going to meet next year in this meeting. So now going forward for the pillar 4, that is again very close to us. It's improving the CAN diagnosis. How can we address the misdiagnosis or undiagnosis for CAN in the community to again increase the numbers, like how Dr. Chung was saying that we need certain number for us to be able to move forward in the clinical trials pathway. So what we are going in that, we're doing little contribution for that aspect of the work as well. And we're going to look at the cerebral palsy and CAN. Now, as we know that roughly about 3 individuals in 12 CAN individuals, they had a misdiagnosis of cerebral palsy at some time during their diagnostic journey. So we have joined hands together with Professor Joseph Gertz and Dr. Claire Van Eek, who are basically studying the genetic causes of cerebral palsy. They're looking beyond the fact which was earlier considered that cerebral palsy is mostly caused due to the perinatal trauma. They want to look at the genes which are responsible for cerebral palsy. So for this particular project that they are doing at their end, they have collected a huge sequencing data from a very well clinically characterized Australian cerebral palsy population. So we reached back to them. We are working very closely with them so that they can go back into their databases and they can go back to the sequencing result and see if they identify any KIF1A individuals or not. So if they identify, they connect with us, and then we connect them with the org, just to facilitate that connection, to facilitate that the improvement in the diagnosis, especially in the cerebral palsy population. Along with that, we are working at the moment just with the Victorian Clinical Genetic Services to encourage them, to help them find the evidence-based studies such that they can include KIF1A gene onto the special genetic panels, which are often requested by the clinicians when there is a suspicion about the genetic disorder based upon the phenotype. So we're working parallelly on that respect as well, so that when any individual is coming for the genetic diagnosis and when a particular gene panel is requested, for example, the CP, now the KIF1A gene is already there on the CP panels in the Australian diagnostic facilities. Now, the next thing that we are working on is the biomarkers for CAND. So biomarkers, they are essentially the indicators of our health and disease, like what's going on within the cell at a particular time. So in order to find those early predictors of the disease, like Dr. Chung is working on the blood samples, we shifted a little bit of gears and we wanted to do things in parallel and see if we can find the biomarkers using other possible ways. And the ways we can think of is, first of all, using the brain organoids. So what we are going to do is we are going to take the brain organoids and we're going to look deeper to find the molecules which get dysregulated up or down in the canned individual as compared to the normal individuals. Now, the pipeline for this particular project has been well established by Associate Professor Wendy Gold, who is looking at other neurodevelopmental disorders, and we will be using the approach to find out the biomarkers for CAND. The next thing that we have done in parallel is the collaboration with Professor Bekim Sadikovic. He's an expert who has looked through many different genetic neurodevelopmental disorders to look for those key signatures, what we call it, as the green dots in this schematic here. So these are the signatures which are very specific for a particular disease, so that if the normal genetic screening is not conclusive, then we can go back and request a special test, and then can look for that does this disorder has got a specific episignature, or what we call as the specific the signature on the gene or not. Now, this has been done for a range of different neurodevelopmental disorders. This hasn't been explored for CAND yet, and Professor Beckham is very happy to collaborate with us to look for if there is a signature there or not. And if we found something from the very prelim studies, then we're going to escalate for more numbers. And once we are confident, then there will be an opportunity to include this, to include KIF1A into the special panels that they are designing, which is called the EpiSign, to look for these signatures, again, as a part of the genetic diagnosis journey for the individuals. So coming to the last pillar, that's raising awareness for CAND. This is one of my favorite slide. Like, you know, when we talk about CAND, my eyes just shine up. So again, like just letting the community know what CAND is. Now, how we want to do it. We want to basically reach out to the news and the media. Unfortunately, there is not much awareness, especially in our part of the world, which is Australia. So we want to, again, make use of the opportunities as they come through to let the community know about KIF1A. Now, the second thing that we have done is we have created fact sheets for clinicians. Again, we worked together with several members of our KIF1A Research Network members to make these fact sheets, which can then be given to the clinicians and the pediatricians so that they are aware about CAN, what to expect if there is an individual coming into the clinic, and if they notice some signs, which is mentioned in the fact sheet, how to go forward with that. So I think reaching the clinicians and the GPs as well, that can help to kind of raise awareness about what CAND is. What we're doing in terms of engaging with the other organizations. So we are joining hands with other organizations who are also working on the rare genetic disorders. For example, the Childhood Dementia Initiative and the Rett Syndrome Association of Australia. It's wonderful. Like they want to support. For example, if you're writing some fundings, they would be very happy to support our funding applications because they know that once we found out something for CAN, it might have a wider applicability to other disorders as well. So it's wonderful to work together in collaboration in that respect. We want to raise funds, which we desperately need to support our ongoing KIF1A work. So we are creating donation pages for CAN research, and we are again letting the community know that yes, if they would like to donate, this is a place and this is how you can do it. Conferences, we find that conferences is a wonderful way again to spread our message across to the community about CAN, and this is where we really want to present in as many conferences as possible. Now, the wonderful thing is that the other organizations, they're acknowledging our relationship with KIF1A.org, which is really powerful. Like they're acknowledging that we are reaching out to families to know about their priorities, to know about their perspectives. And for example, I didn't mean to put my picture here, but this was the only thing that I could get from Google. So I got this 2023 Engagement Award from University of Melbourne solely because of what KIF1A.org is doing, like how they have Thank you. Like this is a beautiful relationship that you had. This is what the person who gave me the award, he said, "Oh, you've created such a beautiful relationship with KIF1A.org. Keep growing, keep moving forward." And it's really special. So yeah, I wanted to put it on in my advocacy slide. And at the same time, we had our first Australian KIF1A family catch-up at Murdoch Children Research Institute. It was so nice to see the families in person, to welcome them and to show them firsthand like how we are doing research in the lab. It really helps us to motivate and keep focused. So if any of you is on the other side of the world again in Australia, please reach out. We will be there to host you, to welcome you, and to be there with you. So with this, yes, I have acknowledged most of the people during my talk, but yes, a huge thank you to Professor John Christodoulou, who is the head of the lab, to give me this opportunity, to give me this independence to explore KIF1A more. And of course, to the travel support funded by University of Melbourne Early Career Academic Conference Support Scheme, as well as KIF1A.org. So with this, and thank you so much to all of you for accepting me in this family. And yes, I'm looking forward to engage with most of you during this weekend. Thank you. And yes, thank you so much for posting these postcards. They're very special and they're always next to my desk. And thank you so much for someone who created this beautiful Christmas tree. Like these little things, I would say they are not little, they are big. They're very, very special. So please, I hope we keep doing and we keep sharing this relationship. Thank you so much. Any questions?
I believe we have time for 1 or 2 questions, and then Sim will be back up here for a panel discussion after the presentations. So.
Then you sort of have like an optic nerve. Have you expressed any of the mutants, KIF1A, and see what happened with the nerve? Do you see any degeneration there?
This is a beautiful question. Thank you so much. Actually, we have found this collaboration very recently, last month. And the good news is that they have taken it on very quickly. So the brain and the eye organoids, they are currently growing. And hopefully once they mature, we start to look at those markers in the optic nerve atrophy and to see if there is any changes which we can replicate in the dish. So yeah, this project is very well going on and it's on the gear.
The microphone is closed. Yeah, so in the molecules that you're trying, the FDA approved, there was one wasn't clear to me. What it was doing is increasing the expression of KIF1A, or it was binding to KIF1A. Do you know what the?
Great question. So when we started this expedition 2 years ago, our focus was to improve the mobility of KIF1A, to again improve its attachment and to improve its walking pattern. Now, how high throughput drug screening work is that we first have to find out some kind of something that works and something that doesn't work. And we spent quite a bit of time to find something that works because we already don't know about this. And this is how like the background Z prime calculations that works for it. So when we were hunting for these things, we come across these tool compounds. That's why we call it as a tool compounds. They're not FDA approved. And we've suddenly found that there are some compounds which is increasing the expression of KIF1A. So we have changed our focus a little bit from finding it out something that improves the velocity to something that improves the expression. We are definitely working on the other side as well at the background, but at the moment, I think we are still going to go ahead and because nobody has explored this further, we do know that if these drugs are working, like how Dr. Chung was saying, we've got 2 copies, one is healthy, one is defective. then if you're putting a drug that can increase the amount of defective protein as well, which is again going to cause toxicity within the cells, we don't know yet, but I think we're working on a hypothesis that, again, variants, they affect the KIF1A function in different ways. And if we can find something which is at least increasing the wild type expression, then hopefully the wild type is going to match up with the other wild type, and that can rescue the function of the mobility. So this is one side that we're working at the moment, but we don't know whether those drugs, like how they're acting. So this is something that we would like to explore further.
Yeah, hi, good morning. I just want to thank you. I met you in 2019, and I think your talk was the one I remembered most. So thank you. You just have
Thank
a
you.
wonderful way of breaking this down and all the work that you're doing for our children. I do have a question. So you're in Australia. What does it look like when you feel like you have a small molecule and you want to get into a clinical trial phase? How does that work with the FDA in the United States or people all over the world? How do we get from the lab to actual clinical trials from your perspective with small molecules?
And again, great question. Thank you. So with the small molecules, I think it just depends upon which way we are going. So if we go ahead doing the synthesis from the start, it's a long process. Like we have to go through again the lab-based studies. We have to convince them it's working, and then we have to look for the safety studies, again showing that it's efficacy, it's safety concerns, and we have to go through with the TGA. So that's the therapeutic good administration in Australia to convince them that this drug is working. That's the reason that we are not going on that part at the moment, because again, it's taking more time. It's going to, we need to have more data to convince the regulatory authorities. So that's why we are working on the FDA approved drugs. So once there is a already FDA approved drug that works on KIF1A, the system is much more simpler. So there is a lot of, because the TGA is very receptive of the drug repurposing in Australia, which is why we want to explore that further. That's definitely, it's a process, which again, we have to do the safety studies, but maybe not in a very detailed way or not in a very big population. So it's much quicker, and we do have support from the organization as well. So once we find a particular drug which is working, then definitely there are teams within the institute which can help us to escalate, which can engage with the regulatory authorities and help us to navigate through that pathway.
Thank you, Sim.
Thank you. Thank you so much.